Great. So hello, everyone, and welcome on the seventh actual session of the first module of Seismic. Okay, I guess you can see my screen now. So as an overview, I've written here, what we are dealing with today is to discuss how we calculate the half-life. Then we are moving into the gene expression and regulation model. And finally, we are moving to the genetic toggle switch. I'm explaining later what this toggle switch is. Going straight to the first topic for today, calculating the half-life. Here we are given some equations. Basically, you can see the notes given to you via the Seismic website. I just copied this into the Matlab code, and I've added some comments as well for you to see it yourself on your own time. However, let me just give you a couple of comments before proceeding to the gene expression model. So the equations given, as you can see here as well, the first equation given is the result of a mathematical analysis of a model. So it's basically the solution to that model. And in our case here is an exponential. As you can see, the exp here means exponential. It's an exponential function. And we've got y of t equals the y naught, which is the initial condition of y, and then it's times the exponential function of minus c times t. T is the time, and c is a parameter that allows you to regulate how steeply the decay goes down, or yeah, because we are talking about half-life. We can use some data to feed the function to the data, to this, to use y to feed the function to the data. But this is not the purpose for this topic here. We are gonna do it anyway in the next section. So here, let's discuss how we calculate the half-life, how we end up with the half-life calculation. So we've got the initial equation, which I explain here what it represents. Let me also give t is the time, and y is the concentration of a protein, for example. So y naught is the initial concentration. Then, in order to end up with this equation here, we need to do some rearrangements in order to find the half-life. This is the point, the time point where the actual concentration of the protein y on the left-hand side, as you can see here, is equal to half the starting population, half of y0. So we are looking to find how long will it take until the function has reached the point of half the population. Starting with 10, for example, when do we reach 5? So what we need to find is the concentration where y is when the time point when y the initial the concentration equals the initial concentration divided by 2, so the half the initial concentration. Then we substitute this to the initial equation that we had, and we end up with this equation here. And then we divide, we do some rearrangements as said. We divide by y0, we end up with that, and then we need to get rid of the exponential. So we add the logarithm in both sides, left and right of the sign equal. So having the logarithm of exponential gets rid of the exponential, but now we've got the logarithm of 1/2, which is again translated into minus log of 2. If you have log of 1/2, you can add the minus on the front, and then you've got logarithm of, and exponential. So we've got on the right hand side minus t c times t half, and rearranging this, we've got t half, so the half time equals the log of 2 over c, and that's how we end up with the half-life. Moving on to the second section for today, the gene expression regulation model. It's an assignment that I copied here. I also added a couple of comments that we are going to discuss now, and then we are going to see how to solve this assignment. So what we need to do is to initially look at the code which models the expression of x regulated by transcription factor y in a system described by the equations below. These are the equations. So we've got dx of the dt, and then the y of dt over dt. And as we can see here, there are two different differential equations. The first one is for population x, and the other is for population y. Talking about gene expression, as described above, they are two proteins that have an interaction with one of the proteins being the transcription factor. Y is the transcription factor that has an impact on the transcription or creation of the other, the X. Transcription factor is Y, as mentioned below, and we are looking at the expression of X. We are given in the exercise some initial conditions. For C should be equal to 0.1, B should be equal to 1, G equal to 1, and the initial concentrations is 0.1 for X and 10 for Y. Then we'll need to modify, but let me come back to this later. So to work this problem, this assignment that I just read through, we need to first find the solution of the equations, and then plot the graph from that equation, the graph of the solution of the equation, and then look at the exponential decay and analyze what do we see in the graph. As said in the past, we always first clear the command window and the workspace. So we use clear and CLC, and then we proceed to the actual assignment. In order to proceed with this problem, we need to create some symbolic equations, and that's why we are using the sims function here in order to add the symbols that we are using in the equation, to define the symbols that we are using in the equation. once we are done with creating the symbolic equations, we need to solve them using the dsolve function that we've seen in the past. And then we need to convert the symbolic variables to algebraic functions, to algebraic variables, and then plot them. Let's take it one by one, step by step. So, as said, because we are dealing with symbolic equations, we first need to define the symbols that are being used in the equations. And as you can see above, what we've got is and y of t of the time. So this is what we need to define first, and that's why we are defining x of t and y of t first. Then we've got the constants, other variables, the other constants, which are c, b, and g. We don't need to assign the values at the moment, but we just need to assign them, that we've got constant c, constant b, and constant g. And that's what we do here, g, b, and c. And then we need x naught and y naught as the initial conditions that we are going to use. And they are here, they are defined here. Again, we don't need to assign them the initial values, but we just need to define them in order to be used in the equations later. Again, x is the protein of interest, the one for which we are looking at this expression, and y is another protein that regulates the output. It regulates x, protein x. Then we need to create the symbolic equations and assign them in a variable. I'm naming this variable differential equations. And as you can see, with plural equations, because we are assigning into this variable two equations, one for x of t and one for y of t. Because we are assigning both these equations into one variable, we need to use the square brackets. So we add all this in square brackets, and we've got differential x of t, which is the x over the t and the y over the t. Then we need to assign to have the right hand side of the equal sign, and assign it into the x over the t and the y over the t. So here we've got gy minus bx, and this is what we've got here given by the assignment. And for the other one, the y over the t is equal to c. Is much easier, that one. So we've got it equal to c. In order to assign it, to use the equal, we use the Boolean equal. I've not added any links at the bottom today, because all the functions that we are using today have been used in previous seminars, in previous calls. So there is no need to add them again. You already visited the site, and the math works, and you already seen how this works. If you don't remember, then please have another look at the MathWorks website. Talking about the Boolean equal is the logical. It's being used mostly for the logical equals, but it's also used here to assign the right-hand side into the left-hand side in an equation. This is commented out because it's gonna be used in the second part of the assignment. So I'm gonna revisit it in a second. I'm now moving forward. Having created the differential equations variable, we proceed to create the initial conditions variable, where we are gonna assign the initial conditions of x and y, which is x0 for x and y for y. Again, this is a symbolic equation. We don't assign any values. So what we need to do is to assign x0 into x naught in the brackets. That means it's at time 0, time equal to 0, because we've got x time equal to 0. It has the concentration x naught, and the same goes for y. Again, we are using the square brackets because we are assigning two different equations into one variable, which is called initial conditions. And then having created the symbolic equations, we need to proceed into solving those equations. And to do that, we will need to use the function dsolve. Again, we've discussed this in the past. So I create a variable named solution, and on that variable I assign the solutions of the differential equations that we've created above. So we've got dsolve as a function, and we use the differential equations and the initial conditions that we've assigned earlier in order to create the solution. And as I said in the previous seminar, always after and dsolve, we use the simplify function in order to simplify the equation that has been created. Let me actually run the code so far. Create the variables, use the dsolve to have the solution. The solution is a structure which we can access here for y and x. So we've got two, it's the y and the x. And then for each of them, we use the simplify to simplify basically the solution. In order to access it, we need to first access solution and then access x or y, which is what I showed you here. Because it's a structure, we first need to access solution and then get into y or into x and simplify the symbolic equation which is within that. So I'm simplifying for x, and I'm assigning it into a variable named x, and the same thing for y. So I will now create two new variables, one named x and the other named y, and assigning to them the simplified version of the solution. As you can see, for x, this is the simplified version. It doesn't look very simplified, but this is the simplest form Matlab can create. And for y, the same. This looks much more simplified. Let's actually start by that one and then proceed to the other. Y, the concentration of Y equals of at any given point, I mean, equals equals to the initial concentration Y naught, which is the initial concentration that we can assign into this equation, plus the constant T times the time. As you can understand, this is a protein whose concentration constantly increases over time. The other one, concentration of protein X, is much more complicated. But what I want you to realize here is that, okay, for the other one, just the concentration, the initial concentration of Y naught is enough to solve this this equation and calculate the concentration of Y. However, for the other protein, for protein X, in order to calculate the concentration, we need both the initial concentration of X and the initial concentration of Y. So we need to know both the initial concentrations of these proteins. What else we need to know is all the other constant values that we've got. So you can see here we are using B, we are using T, we are using C, we are using G. So everything that has been assigned here needs to be used in order to calculate the concentration of X. Sorry, we were here. The expected output that I've got here is commented out. It's not the one we've got here. It's the one we should expect to get when we've got the differential Y over T equals to minus C times Y. It's the one that we are gonna see in a second for this second part of the assignment. But now we are here. We've just simplified the symbolic equations for X and Y. We've got, this is what we've got here. And the next step is to convert the solution of Y and X to an algebraic function. Basically, from a symbolic variable, we are converting it to an algebraic variable. And to do that, we'll need the Matlab function. This is a function we've dealt with in the two previous seminars, so have a look if you don't remember them. And I've copied above the symbolic equations just to see the symbols that we are using to be easier for us to complete the Matlab function. What I'm doing here is to assigning into a variable named matfun for Matlab function Y. That's for equation Y. We are using the Matlab function as set for Y, and then we are assigning the variables using the inverted commas, and in brackets we assign the actual variables. As you can see here, I'm using T, Y naught, and C because this is what we've got in the symbolic equation. We've got T, C, and Y naught. We've assigned everything here. And then let me first just run this part of the code, and then we need to solve actually this equation the algebraic equation by assigning values into the variables now, into the constant. For example, here we've been asked, and this has been copied from the assignment. It's found here. I just copied it below for us to be easier to see what we need to assign into the into the algebraic variable named matfun y. As you can see here, we've got C needs to be equal to 0.1, B needs to be equal to 1, G needs to be equal to 1, and the initial concentrations are for x not equal to 0.1 and y not, the initial y concentration is equal to 10. For y, we only need the y naught, which is 10, as you can see here, 10, and the c, which is equal to 0.1, and this is what we've assigned here. T remains as t because this is what we are solving for. We are solving for t. So I'm running this part of the code as well, and then we need to plot it. I'm assigning some random limits. I initially set it to 0, from 0 to 50. It was fine, and I kept it there. Otherwise, I would need to increase or decrease the limits based on what we need to emphasize on in the plot. So limits 0 to 50 are just fine for this situation, for this exercise here. And what we do in order to create the plot, we first create a new figure. So I just created an empty figure, and then we use the f plot in order to plot the fy1. This is the variable I've assigned the solution of the algebraic function. So we are plotting fy1 using the limits I've assigned above, and I'm just changing or creating the color of the line. The line width is set to 2, so it's a bit wider in order to be easier to spot. And then let's run this first. So as said, it's a straight line, increasing, constantly increasing, because the equation is y naught. So the initial concentration starting from 10, because we've assigned number 10 as the value for the initial concentration of y, and then for each time point this concentration increases. You can see it's a plus of the initial concentration by the time times the constant c, and that's why we've got this shape of the plot. This, it's not a curve, it's a straight line. Let me also add the x and y labels and the legend, so we can discuss again. In the legend, I've added that this line has the constant c equal to 1. We've got time in the x-axis and the protein concentration in the y-axis. Starting from 10, let me actually show that. Starting from 10, it constantly increases as discussed earlier, because that's the equation. Then we do the same thing for we do the same thing for the second equation for the protein X. However, there is no need to do this at the moment to save some time. Let me proceed to the second part of the assignment, where we will need to modify the MATLAB code to solve the differential equations having the concentration of y described by the y over the t equal to minus c times y this time, and the initial conditions are still the same. So we've got, let me clear it in, we have to assign the same symbols for the symbolic equation because nothing has changed. We are still using c and y and the others that we've assigned earlier. So we assign those symbols first, and then we create the differential equations. For x is the same, but for y this time, instead of c, we've got minus c times y. This is what the exercise asks. So here, instead of running the first equation that we ran earlier, I'm gonna have the second equation. And yeah, we created the differential equations variable having x and y. Y is now equal to minus c times y. The initial condition name as described in the assignment. The solution is being created using the dsolve again, and then we use the simplify function to simplify the algebraic, the symbolic, equations. And these are the expected outcomes, by the way, as I said earlier. And indeed, these are the outcomes that we have. This is a bit less, kind of less complex than the one we had earlier, but it's still complex. Anyway, we don't really care about that because we are not solving it by hand. Matlab does all the work for us. And again, we are moving into converting the symbolic variables into algebraic variables. That's why we are using the Matlab function and then solving it using the real values. And having that, we need to plot it later. So again, I'm using the same limits for now, and I'm creating a new figure and plotting it. And this is what we've got now. Oh, sorry. Yeah, I had to close the initial. This is the equation that we had earlier. Let me close that and run it again. This is what we've got. For y equals to for the y over the t equal to minus c times y, this is what we have. We've solved it and we ended up with y naught, starting with y naught times exponential of minus c times t. This is the curve that we get, starting with an initial concentration. It keeps decreasing at a higher pace initially, so the slope is steeper, and then it starts getting a lower slope, a smaller slope, until it reaches 0. That was for y, and now we need to also convert the solution x to an algebraic function in order to plot x as well. I'm then solving it with using the variables that were mentioned earlier. As you can see here, the difference earlier for y, we only had t, y naught, and c. This time for x, because this is the equation we are dealing with, we need to assign all the variables that are found in this equation. In this case, we've got y naught, it's here, we've got g, it's here, we've got c, we've added it here, t, here, b, and x naught. Here's x naught, and we've added it here as well. And then for each of those, we assign their values as given by the exercise. Here are the values given. So t remains as it is because we are solving for t. And then we've got 1 for b. We have been set to use 1 for b, 0.1 for x naught, as you can see here, 0.1 for c, 1 for g, and 10 for y naught. Solving for that, again, having the limits from 0 to 50, and then I haven't deleted this this plot. I'm gonna use hold on in order to plot the concentration of x in the same plot. So I'm using hold on, and then f plot. In order to plot it, I'm just changing the color from black to red. So I'm using here limits, and the equation has been plotted here. We can minimize the limits, but for now it's fine. But let me have the y label and the x label and the legend in order to be easier for us to realize what we are looking at. So what we've got here is the protein concentration in the y-axis and the time in the x-axis. So as the time goes on, the concentration of, we've got two proteins. The curves represent the concentration of two proteins, the x and y. We've got x in the red color and y in the black color. As said earlier, as the time goes on, the concentration of y starts decreasing, starting from 10 as initial concentration. It decreases as the time goes on, while for x, we've got an initial increase, with a very high slope, a very quick increase, and then when it reaches a point, it starts decreasing together with y. This makes sense because, as we said, y is the transcription factor, and x is the protein for which we are interested in its synthesis, in its transcription. Initially, the protein concentration is increased. So there's a synthesis of this protein. The transcription factor goes down by that point, and then there's a decrease of that protein again. Both the newly synthesized protein and the transcription factor as well, they keep decreasing. So that's a decay process until they reach 0. This is what we can see in this plot. And I've added here a comment that we can use the model. Let me close that as well. We can use the model to see what the different predictions regarding the half-life are. We can use this model to add different numbers for the initial conditions. I mean, the C, B, G, and the initial concentrations of the two proteins. And that way we can study in detail the behavior on the model. And we can, for example, compare to an experimental situation. I mean, the rise or the decay, the half-life, and so on, and see if they are in agreement with the data that we get from the experimental situation. Let me move fast to the third part of today's seminar, because we don't have enough time. We are talking about the genetic toggle switch, and as promised earlier, I'm going to explain what this is. It's basically a model that shows how to create bistability using simple assumptions about gene expression, which has application in synthetic biology. It is an approach, but it's not just about synthesizing something. It's an approach about which basically bases synthesizing on rational design. And by saying rational design, we mean that we make a specific model based on what we think it goes on, and then we create the experimental situations. We do the experiment to see what really happens, and then have the comparison to see if our model fits what really happens. And for example, we can use this approach, the genetic toggle switch, to make predictions about the coronavirus, about some random pandemic or whatever. I've copied the assignment below. Where we should model a toggle system. We've been given these equations to use. So we've got alpha, they are random equations. And I guess there are two neurons that have contact with each other. We use alpha 1 here and alpha 2 here over the exponential of beta times v and gamma times u minus u or v in each of the two equations. So it's the same thing as we did earlier, but with some differences. In order to proceed with that one, let me first clear the window, the command window and the workspace. In order to proceed with that one, we need to have a function. I've already created this function named under toggle model. That's the function's name. And these, by the way, are given in the Seismic website. I just copied them here in order to be easier. So we've got the two equations, and I've assigned the values into the variables, alpha 1, which is here, alpha 2, which is below, beta and gamma, which are the variables that are being used, or constants, you can say, because we've assigned the values here. And then we've got y dots, which is the output of the function, and we assign the u and then the v, the two variables that we just created by solving using these constants. Getting back to that, by the way, I've added in this in the notes as well. I've added the function. I've commented it out. It's at the end of the notes file. You can see it on your own time later. So we first clear everything. We assign alpha 1, the value 10 to alpha 1, and the value 3 into the beta. We are solving for v. So v is what we are looking for. Alpha 1 is divided by exponential beta, and beta times v. I am assigning this algebraic function into the variable named modified function. We've solved for that one, and then we need to plot. It's again the same thing as we did earlier, and we need to plot it using fplot again, using the limits from 0 to 3, and the line width set to 2, just to have it a bit more wider and easier to view. So this is the, this is how the plot looks like for the exponential decay. Starts from 10 and goes down near to 0. It's similar to what we discussed earlier in the previous plot, so I'm not gonna get more time here. What we are asked is to modify the Matlab code to numerically integrate the equations alpha 1 and alpha 2 equal to 10, beta and gamma equal to 3. This is what we've added here in the function. Then we need to plot the concentrations of u and v. When u has v0 equal to 0.1, u0, sorry, and v0 equal to 0.1 and 1, and for the second is equal to 5 and 4. So what we've got here is again to set the time span from 0 to 10. We are running setting the y0 settings equal to 0.1 and 1 as being set to do here. And what we do is to use the ODE45 function to solve the function that we created earlier, the toggle mod. So we are using the toggle modified toggle function to solve for the time span 0 to 10 for y naught as set above. And then we plot the outputs of this function t and y, as you can see here. Let me have this. By the way, I'm using the subplot, so we've got 2 columns, 2 rows, 1 column, and we are first dealing with the first one on top, and then I'm gonna deal with the second one, the one on the bottom, just to have them one above the other and be easier for us to compare. So this is what we have. Sorry, I should have assigned time span first. I didn't do it earlier. So this is what we've got for the first one. The initial concentration increases while the other one decreases. It decreases, you cannot realize it here because it's very small. The difference is very small, but if I manage to zoom, you can see that it starts from 0.1 and it goes down to 0. While there's an initial increase, a constant increase for the other concentration until it reaches 10. The same thing can go for the second sum plot, having different initial conditions. This time we've got 5 and 4. Again, we use the OD45 to solve the function, where we have alpha 1 and 2 equal to 10 and beta and gamma equal to 3. These are the equations that we are solving for, and the initial conditions that we use are those this time. So instead of starting from 0.1 and 1, it starts from 5 and 4. And this is what we get. It starts higher than the previous time. It goes down and then it increases again, while the Y2 decreases until it reaches 0. As a general rule, the one which starts higher goes higher, and the other which starts lower goes down. It decreases, goes near to 0. What I want to discuss here is that the phenotypes depend on the initial conditions. So by changing the initial condition, basically having the same underlying model, so having the same value equations here without changing these equations, but just changing the initial conditions, as you can see, we've got different concentrations, different variables different concentrations of the proteins, and this means that we will have different phenotypes. So depending on the initial conditions, we end up having different phenotypes, and if we need to change anything on the end phenotypes, we change the initial conditions. This is the comment that I wanted to make here, and I'm stopping it here. Have a look at the on your own time. Have a look at the notes that will be shared with you right after the end of this session, and let us know if you have any questions. But the notes here, the script is very descriptive, so I guess it will be very straightforward for you to understand what is going on. Thanks for your time. Let us know if you have any other questions.